I'm going to introduce our chairperson, Dr. Story Landis. She's the former director of the National Institutes of Neurological Disorders and Stroke and a co-founder of the Brain Initiative, which was formed by President Obama. She's a former faculty member at Harvard and at Case Western Reserve, and her own research has been in the area of neurological development and the role of neurotrophins. So, Story, I'm going to let you chair the session from now on. Thank you.
Thank you very much. I'm delighted to be here. It's my first Galien forum, and I have to say I have learned an incredible amount. I am glad that all of you are staying for this talk because I think we have a beautiful success story to tell you. Not just charting a path, but actually making real clinical progress in SMA, spinal muscular atrophy, which is a devastating disease. In fact, it's the most common genetic cause of death in the first year. If I could have the first slide. In 1995, first slide. In 1995, the gene responsible for spinal muscular atrophy was discovered. It's named survival of motor neurons, or SMN, and it encodes a protein which is absolutely required for the maintenance of motor neurons, which control muscle contraction. And if your muscles don't contract, you can't breathe, you can't move, and the infants die. Scientists soon discovered a second gene, the Smn2 gene, which does not cause the disease. It's a duplicate of the first gene, but it's not an exact duplicate. In fact, it is not competent to make much full-length SMN protein, and you can see there's only one of those lovely squiggles and not the three in the SMN1. Now, if we go to the next slide, if you have SMA, you have a complete loss of function of the SMN1 gene. That is, no SMN protein gets made from that gene, and you are completely reliant on the protein that's produced by the Smn2 gene, which is not nearly enough to maintain the motor neuron function. So in NINDS, when I was the director, we looked at this comparison, no SMN1 protein product, and the possibility of actually getting SMN protein made by the second gene. We thought this offered real potential for developing a therapy. I have to say we were a little naive. We hadn't done much translational research, but we were very interested in learning more, so we decided that we would make SMA the prototype for a novel drug discovery protein in NINDS. Now, started that in 2003. What we found was that we did not actually make a viable clinical product that could be taken into patients. It was a hard lesson to learn, but we discovered that we couldn't. But what it did, which I think was maybe even as positive, was it called attention to the therapeutic possibilities for SMA. Now, that potential was actually met in December 2016, when the FDA approved the first treatment for spinal muscular atrophy, nusinersen, which is an antisense oligonucleotide, which is marketed by Biogen as Spinraza. And I would just say, for many people who do therapeutics development or study neurogenetic diseases, there has been this notion if you could get the gene, you would immediately have a therapy. Well, it turns out it was 21 years between the discovery of the SMN1 gene and the development of a therapy, and that's actually much quicker than, for example, Huntington's disease or Duchenne's muscular dystrophy. So what we will do in the panel is we will share the story of the successful development of Spinraza. And it's a story of exceptional collaboration between outstanding basic scientists extraordinary clinicians, and 2 companies, Ionis and Biogen, progressing from a mechanistic understanding to design of a therapeutic, to preclinical testing, to clinical studies, and approval. Now, the work that the panel is going to describe was aided by a number of people who aren't sitting up here, NINDS, other NIH institutes like NIGMS, Muscular Dystrophy Association, and Cure SMA. But I want to shout out especially to the SMA Foundation for its essential contributions. The foundation was founded in 2003 by Denneker Singh and Lauren Eng, who are in the audience, and they had a number of goals: raise awareness, increase federal funding. And I can tell you, at NIH and NINDS, every time there was an appropriations hearing, we fully expected to get a question about SMA funding. It was good, reminded us the importance of this disease. They also wanted to recruit outstanding new scientists to the field of SMA. And these are things that many of the foundations, disease foundations, do. But what I think has been very special about the SMA Foundation is that they have engaged with laser-like focus the interest of pharmaceutical and biotechnology companies, and have worked incredibly hard to identify and eliminate obstacles to drug development, making it as easy as possible for companies to actually produce a drug. Funding biomarker development, creating tools, natural history studies, and clinical networks. And I know they're here in the audience, over there. Raise your hands. Thanks. So the members of the panel are Dr. Darryl DeVivo, Sidney Carter Professor of Neurology and Pediatrics at Columbia University Medical Center, directs the Pediatric SMA Clinical Research Center and the Pediatric Neuromuscular Research Network, which was used for SMA clinical trials. And both of these were funded by the SMA Foundation from their inception, and I don't want that up yet. And then Adrian Crainer is the Professor and Chair of Cancer and Molecular Biology at Cold Spring Harbor Laboratories, and is an expert on the mechanisms of RNA splicing, the editing of RNA messages copied from our genes, and you will understand very shortly why that's so important. One of those outstanding investigators attracted to the SMA field. Dr. Frank Bennett is Senior Vice President of Research at Ionis Pharmaceuticals, a founding member of the company, and this company is focused on the use of antisense nucleotides as therapeutic agents. Kathie Bishop was at Ionis Pharmaceuticals, where she was the VP Clinical Development and leader of the development team for Spinraza. And Dr. Al Sandrock is the Executive Vice President and Chief Medical Officer at Biogen. So this should be a very wonderful talk about a real success for kids and families. Darryl.
Thank you very much, Dory. And my goal here is to introduce you to this disease. It's pretty a devastating disease. Story has described it to you in words, and I'm hoping that I can show it to you in a video. So let's see here. Okay. So you can see here 3 children, ranging from an infant lying quietly on her back, and then in the middle, an infant just barely able to sit and maintain her posture in the sitting position. And then to the right, 2 patients. This is a young boy who is able to move about independently. He struggles to get up from the ground, what's called a Gowers maneuver, indicating proximal weakness. And then this young girl who has a similar difficulty in ambulating. So these 3 phenotypes, type 1, type 2, and type 3, embrace about 99% of all the patients with SMA. All of them have the same disease-causing mutation affecting the Smn2 gene that Dr. Landis just described to you in some detail. But the number of Smn2 copies vary considerably from one patient to the other. So the more copies you have, the less severe the phenotype. the less copies you have, the more severe the phenotype. So the type 1 babies, as a rule, have about 2 copies. The type 2 patients, as a rule, have about 3 copies of the SMN2 gene, and the type 3 patients have usually 3 or 4 copies of the SMN2 gene. So the more the copies, the less severe the clinical phenotype. Now, one of the unfortunate issues among many here in this devastating disease is the fact that the most severe phenotype is the most common. About 60% of the patients who have spinal muscular atrophy have the most severe phenotype. As a result, that contributes to the point that Dr. Landis brought up earlier. This is the #1 genetic cause of death in infancy. And the reason for that, in large part, is the fact that 60% of all the babies born with SMA have the most severe phenotype, and therefore they are at great risk of dying within the first 2 years of life. Within the first 10 months of life, they will lose strength in their breathing efforts so that they will have to have permanent ventilation. They have difficulty chewing and swallowing, so they'll have to have nutrition provided through a gastrostomy tube. And also because of the associated weakness of the muscles, they have difficulty maintaining their musculoskeletal system. So they develop deformities of the joints, curvature of the spine, weakness of the pelvis, difficulties with the hip joints, and as a result, that contributes increasingly to disability and to pain and suffering that is associated with this disease. So it truly is a devastating disease. It's one that represented a huge unmet need until recently, and the more of that story will be heard shortly.
Yes.
Adrian.
Hello. So I'm Adrian Crainer. I'm an RNA splicing researcher, and I've worked at Cold Spring Harbor Lab on Long Island for the last 31 years. And so I'm one of the inventors of nusinersen, or Spinraza, and worked on its preclinical development in a close collaboration with Ionis Pharmaceuticals. This all started with a very solid foundation of basic science, going as far back as 40 years ago, pretty much exactly, when Robertson Sharp discovered that our genes are made up of exons and introns, and that the introns are removed and the exons are joined at the level of RNA splicing. So Spinraza is the first approved drug that directly targets and corrects a splicing defect, and it does so at the RNA level. So you've already heard that the gene, SMN1, was discovered by Malky and colleagues in 1995. And let me see how I can point. So what's critical in patients is that they have a remaining SMN2 gene despite the loss of the SMN1 gene. And so both of these genes are nearly identical. However, there's a critical difference that was reported by Lorson and colleagues in 1999, which is in exon 7. There's a C in exon 7 in SMN1, but it becomes a T in SMN2. And the net result of that is that the spliced messenger RNAs from SMN2, for the most part, lack exon 7. So you get, as a result, a truncated protein that doesn't hang around very long, very unstable. A small amount still include exon 7, and its a key feature is that the C to T change doesn't, in and of itself, change the amino acid that's encoded, as long as the exon is still kept in the messenger RNA. So SMN2 can make full-length SMN protein, but at low levels because of this defect in splicing. So what we worked on is a method to convince this gene to utilize exon 7 more efficiently during RNA splicing. So we were, during the 90s, my lab was one of several doing basic research to identify how exons are recognized in general. What are the RNA binding proteins that govern that decision to skip or include an exon? And so we identified and characterized a number of those proteins. And after the discovery of the C to T importance in 1999, so we shortly after we began to work on the mechanism. What is it about this C to T change that causes exon skipping? So we worked on that for a couple of years and then began to explore ways in which that deficit in splicing of this transcript could be corrected. And in 2004, we began a long-term collaboration with Ionis Pharmaceuticals, specifically to search for antisense oligonucleotides that would promote exon 7 inclusion. And so many oligonucleotides had to be screened. The premise is that an oligo could block the binding site for an RNA binding protein that functions as a splicing repressor. And we found a number of these, but the nusinersen is shown here. This is its actual sequence. It's an 18 residue antisense oligonucleotide. It has perfect complementarity to only one sequence in the human genome, namely this one, in intron 7 of the Smn2 gene. And we were able to show with Ionis that when the oligonucleotide binds here, it prevents these one of these two repressor molecules from recognizing a motif that's also present here. These are repressors of splicing. The fact that we're blocking them means that now exon 7 is recognized more efficiently because there's another key factor called U1 SNRP that binds here to the beginning of the intron. So that's the mechanism of action. And so this molecule, the precise modifications present in it and its sequence, we published that work with Ionis in 2008 in American Journal of Human Genetics, and also described the mechanism of action. And we also showed that it could engage its target in mouse tissues using mouse models of spinal muscular atrophy. We then had to work on figuring out a way to deliver this molecule to the central nervous system so that it could act on splicing in motor neurons. And together with Ionis, we were able to do that in the mouse models of SMA. And this had, it led to striking improvements in phenotype, in survival, and motor function. And with that data, it took several years, but then Ionis was in a position to launch full clinical development. So before I cede the podium to Frank Bennett from Ionis, I want to thank funding agencies and organizations that permitted this work. And when we first started to study the basic mechanisms of SMN2 splicing, we got seed funding from Fight SMA, and shortly after from NINDS, at the time when Story Landis was the director. But at a critical juncture, when we began the screening for oligonucleotides with Ionis Pharmaceuticals, we had generous funding from the SMA Foundation. Thank you. And another pivotal point was figuring out how to deliver this antisense oligo to the central nervous system in mice, and then we had funding from the Muscular Dystrophy Association. Other funding for SMA research was from Cure SMA and from Ionis itself. I want to go back to the theme of basic research. I've had support in my lab at Cold Spring Harbor from the National Institute of General Medical Sciences continuously since 1989, and that's to understand how RNA splicing works. And I remind you, it's 40 years since its discovery. We finally have a drug that can correct the defect in it, and hopefully there will be many more like that. And finally, I want to thank Yimin Hua, a postdoctoral researcher who did much of the work in preclinical development while he was in my lab. Thank you.
Thank you very much. Frank.
So first off, I'd like to thank Story for giving me the opportunity to speak here today. And secondly, I'd like to make 2 points in my discussion. The one is how the biomedical research ecosystem really came together to make this drug happen. And it started with the investments in basic science that NIH and other agencies have been doing for the last 60 years or so. That really created the fundamental foundation upon which both the drug and the mechanism of the drug was based, as well as the technology platform that we utilize. And then secondly, the foundations, the various foundations that were involved were really critical for us in making the transition from really a basic research project to beginning to think about clinical programs. And the SMA Foundation was essential in helping to connect us to key opinion leaders like Dr. Vivo, who really guided us in thinking about how to do clinical studies, as well as funding work that was important for de-risking the project. They funded a large natural history study that I would encourage any foundation to really think about funding, because that was critical. We knew what the natural history of this disease was that allowed us to design what a clinical program was like. And they also funded work identifying biomarkers that could be used to help support drug discovery, as well as being good colleagues and friends throughout the process. Also, I'd like to acknowledge not only basic researchers, but clinical researchers that gave willingly all their time that we needed. And it's one of the few projects I've ever worked on in my life where I can call a very busy physician like Dr. Vivo, and he's willing to give me all the time I needed and not ask anything in return. And actually, his dedication to the patients is really inspirational, and I think that's what drove us to going forward with this project. And finally, my colleagues at Ionis and Biogen for going forward. And then finally, I should acknowledge the parents and the patients of this disease that really gave their time and allowed their children to be tested with a novel, innovative therapy at a time when there was a lot of risk associated with it. So with that, then, what I'd like to do is just move on to how we translated the data that Adrian had produced in this lab, which is showing that we had a very marked effect in the mouse models of SMA, and begin thinking about how we could translate to the clinic. And unfortunately, there is no other species that has this Smn2 gene that was shown on the previous slide, if we can go back one. Other than humans. And so there's no way to do any preclinical pharmacology in trying to establish doses other than in the human situation. So fortunately, there are several mouse models that express this gene as a transgene, and we're able to use the mouse models to help identify what doses were producing therapeutic effects in the mice and the duration of action. And as Adrian mentioned, this drug had a very long duration of action in the CNS. A single injection was producing effects up to a year after that single injection in mouse brain. And the drug half-life is roughly about 6 months in mice. And so we use that to leverage to go into larger species, non-human primates. And one of the things that we explored in non-human primates is how to give the drug. And we very quickly identified that a bolus injection, just a 2 to 5 minute injection, was a much more efficient way to deliver the drug to the target tissues than continued infusions. It also made it easier to think about the clinical program because we weren't having to implant catheters and pumps, et cetera, into the small infants. And so it really streamlined the clinical process, discovering that. And so basically what we were able to do is take the data from the mouse model, calculate what the effective tissue concentrations of the drug were that were producing effects, go to juvenile non-human primates, and based upon the non-human primate data, then do modeling to predict the human doses. And ultimately it led to the design of the clinical programs. And I should mention that we felt it was very important when we started this program. You're always balancing safety with efficacy, but given that this was an invasive procedure, it's given by a lumbar puncture, we felt it was important to start the program and start the dosing at a dose that had some potential of being therapeutically effective. It was on the low end of the therapeutic effect, but we felt it was really important for that. And my colleague Kathie Bishop and I spent a lot of time working on that, and we'll let her describe in more detail the clinical programs. And so with that, I turn it over to Kathie.
Thank you.
Thanks, Frank. So I'm going to cover the clinical development of Spinraza. I'm currently Chief Scientific Officer of a different company, Otonomy, developing targeted drugs for hearing loss. But for 6 years, I was at Ionis and worked very closely with Frank on the development of Spinraza. I was head of clinical development, but I also played a second role, which I led the project development team that developed the drug. I'm going to echo Frank. When we started out thinking about Can you go to the clinical development slide? When we started thinking about first going into patients, there were a lot of challenges in front of us, and these were both technical as well as clinical. On the technical side, as Frank mentioned, we'd worked out that the best way to deliver the drug was through an intrathecal injection. However, these are very ill infants, and the children also. What? Oh, advance one more. I do it?
Down.
You can do it.
Thank you. We'd work out intrathecal injection was the best way to deliver the drug. However, there was a lot of nervousness about doing intrathecal injections in SMA infants and children. As you saw in the video, they are severely affected. The infants have severe respiratory compromise, and that's what they die from eventually within about a year to 18 months of age. So people were nervous about doing an intrathecal injection there. And then the children, in addition, have respiratory compromise, and sedation is required for intrathecal injections in children. So that was one challenge. As Frank mentioned, we were also working out, this was the first Antisense drug ever delivered intrathecally in children. Prior to this, Ionis and myself and Frank had conducted a single-dose clinical trial in SOD1 ALS patients, so a specific genetic form of ALS where we developed Antisense against SOD1. That trial was different, though. It was in adults, it was an infusion, and it was just a single dose over 12 hours. So we were really charting new ground here. So we had to figure out what doses to use and also how often to dose, and that sort of evolved as we went along. In addition, this was a really novel technology, so there was a lot of education required with regulatory agencies, including this drug was developed globally, so the FDA, the EMA, Japan. We spent a lot of time on antisense education and splicing education, and that must have been fun, right? And then also at the individual institutions with their regulatory agencies, the IRBs, or in Europe, the REBs. On the clinical side, the challenges were no company had ever developed a drug in SMA, and actually no company had conducted safety or efficacy trials to this point. There were natural history studies, which I'll talk about a little bit, that were really critical and instrumental in forging the path, but no real drug trials or drug development. So there was uncertainty about which patients should you treat. As you saw, there's extreme heterogeneity of diseases. There are the three types. But even within the types, patients are all different. It depends somewhat on their copy number, but it's not exactly correlated. It also depends on where they are in their disease progression, their age, and their developmental stage. So it was a challenge on that side. And there was also no regulatory path forged as far as what would a registration clinical endpoint be, or what should we be measuring in these trials. So the way we overcome those challenges, I'll echo Frank, is really a close collaboration and an extremely collaborative project. And that definitely involved the SMA Foundation, which was critical to the development of this drug. It involved close collaboration with clinicians who gave so generously of their time to help us figure out how to do this trial, and had a close working relationship with them. It involved a fantastic team at Ionis that really dedicated so much. Frank, but the rest of the team were so dedicated to this project. And then, just as we were starting clinical trials, it also involved the involvement of Biogen, which was a fantastic partner for this project. And the way the partnership worked is Ionis did all the development work, but Biogen was the partner on their project, and they would take over at registration and commercial stage, and Al's going to talk about that. I think that partnership for the two companies really speaks to the strength of having a novel biotech company who really knows their technology and is willing to forge this forward, with the added strength of our larger company who brings a lot to the table. One, to be honest, is financing the project. It was nice every year. You make decisions at a biotech company about what's going to get funded and what's not. Once they came on board, this was always funded, and it just took that pressure off. In addition, they bring the expertise of being a commercial partner, knowing what it takes to get a drug paid for, which I think everyone was just talking about, not only registered, and bring that broader experience that you don't have a smaller company. So I think it was a very strong partnership. So clinical development, this is just an overview. It started in 2012. The drug was approved, as Story mentioned, in late 2015. Did I get that right? 16. Thank you. So it's exactly 5 years from the time the first patient was dosed until the drug was approved. And the drug's now been approved not only in the US, but Europe, Japan, and many other places to come, which is remarkably fast, especially considering that this is a progressive neurodegenerative disease where your phase 3 trials require lengthy trials. Development, though, I will say, and setting the stage for development really started well before 2012. So the SMA Foundation in 2014 formed a network of clinicians called the PNCR. And that PNCR network, which consisted of Darryl and his team at Columbia Medical University Center, Richard Finkel, who originally was at CHOP in Philadelphia, but is now at Nemours Children's Hospital in Orlando, and Basil Darris, who's at Boston Children's University. They did an enormous amount of work doing natural history studies in the infants and the children. They published that work, which was very helpful to us. And they also gave us all the data from those studies, which was instrumental in us determining how to do the clinical trials, especially the phase 3 trials. What this network did for us also is it established 3 clinical trial sites that really knew how to do trials in SMA, which de-risked the project for us enormously. So it's not only the 3 clinicians that I mentioned, but their staff and their teams, coordinators, who are sort of the frontline people dealing with patients, and their parents and their families knew how to do trials, and they shared that information with us to help us along. And then on top of that, the clinical outcome measures in SMA aren't done by Darryl or clinicians. They're done by physical therapists, research physical therapists. And we are very lucky, especially to have Jackie Montez at Columbia University, who, along with Darryl and his team and the network, had developed outcome measures with funding from the SMA Foundation. They developed outcome measures in SMA. They developed manuals, and Jackie really led the training during all the trials, which is critically important to collecting good data on these types of outcome measures. So with all that setting the stage, we started clinical development in 2012. The first trial, as Frank mentioned, we wanted to start at what we hoped might be an efficacious dose. But the challenge here is that we were doing drug development in a really critically severe illness, but having to follow, of course, all the standards of the FDA as far as looking at safety, starting at a lower dose that they felt and then working your way up to where you want to go. So we carefully monitored safety in that trial. That trial was a single-dose trial, standard dose escalation. We decided to start that trial in children with SMA, and that was a conscious decision not to start in the critically ill infants. It's not reassuring when you hear from the field that previous trials in infants with SMA, one done by a network called AmSmart, had been unable to be completed because all the children got hospitalized and they couldn't come for visits. It's also not reassuring when you hear about a natural history study, another one conducted where they flew the infants, and because they decompensate at altitude due to their respiratory illness, they had to be rushed off the plane to an ambulance. So that made us very nervous about starting in infants with SMA, especially when we were really required to establish safety in these early trials. The clinicians also really helped us establish how to do the intrathecal injections in children. I'm going to disagree with Frank and say it's not an invasive procedure. It's a fairly simple injection, but the trick here is doing it in infants and children with respiratory compromise. And the clinicians at the site, and especially another person, Manon Hesh, who's a anesthesiologist at Columbia University Medical Center, helped us work out that procedure. So that trial was successful. We showed we could do the intrathecal injections with SMA. There were no safety concerns coming out of that trial. And additionally, at the higher doses in that trial, we thought we started to see signs of clinical efficacy on an outcome measure that measures motor function in these children. But it was an open label trial, so you always have to be cautious. Following that, actually for all the trials, all along the way, all the children or infants in the trial got extended dosing. So they were dosed throughout the development in open label trials. And data from those open label trials was really critical because it showed the longer you treat, the greater amount of improvement you see. It might take some time in the children and infants to see improvement. And they also, every time we did injection, we could collect CSF fluid, which gave us important information to verify that the half-life was as long as Frank said, and we'd seen in mice at least 6 months, and critical information to move the program forward. So following that, we started a multiple dose trial in children with SMA. This was the first time multiple doses of an antisense had ever been given. Similarly, we saw good safety. Intrathecal injections all went well. And again, we thought we saw signs of efficacy, but again, it was an open label trial. To that trial, we also added an additional higher dose, 12 milligrams, which ended up being the registration dose. So as you can see, overlapping with that trial is an open label trial in infants. So once we felt comfortable about the safety of multiple doses and we got into the 12 milligrams, we felt comfortable enough to start an infant trial. And so that infant trial in the beginning, they got multiple doses, then they did good, so we just kept extending the trial and making it longer and longer. So they ended up getting dosed once every 4 months for several years, collecting open label data that Al is going to show you. There were a couple of critical things that came out of that trial. One, again, that we could do intrathecal injections in infants. We also scaled the dose because of their smaller brain size and CSF volume in that trial, and the PK data confirmed that that was the correct way to dose. We also, from that trial, in careful examination of the data, developed a novel endpoint looking at motor milestone improvement of those infants, and that was something that was discovered as the trial went on, and that endpoint ended up being the registration endpoint in the infants. And then as well, unfortunately, early on in the trial, early in dosing, 3 of the infants in the study died. And through the work of Richard Finkel and myself, we were able to get tissue from those autopsies. And that data from that analysis was really critical for us feeling much more comfortable that this open label clinical improvement we're seeing was real. So analysis of that tissue showed that, yes, we were getting antisense drug to the spinal cord, and we were actually getting it to motor neurons where it needed to be. We also looked at splicing changes, like Adrian showed on his slide, in those infants, and we saw exactly the splicing changes we would expect to see. And then in addition, we used immunohistochemistry for SMN protein, and so the final step is we saw an increase in SMN protein in those infants compared to some control infants that Charlotte Sumner at Hopkins, I think, very generously donated to us for the program. So with that said, we felt comfortable moving into the phase 3 trials, which Al is going to describe. Those phase 3 trials, though, were designed based on the data from the original trials and also a consultation with the FDA, which around this time became, I think, very supportive of the program and really helped with the design of the phase 3 trials, especially a novel interim analysis that we included in those trials, which really was successful in both cases and allowed patients to get access to the drug, and drug approval happened faster. Before I turn it over to Al, I will mention one more thing. There's another study on there at the bottom, Embrace. Another thing that I want to talk about is the patients and especially their parents. So we had enormous support from parents through the study. We did a lot to educate the SMA community on the drug development process. But this is an area where I really feel strongly as a drug development community, we still need to do a lot more work, especially around expanded access during trials. As you can imagine, with these critically ill infants, we had enormous pressure to give the drug out to whoever wanted it during the drug development process. And we did not do that, both for ethical reasons and we didn't want to risk, in the end, getting the drug approved. But I don't feel that the process for dealing with that issue works very well currently. Okay, thank you, Al.
Thank you very much. Al?
Well, thank you. I want to second everything that Frank said about the ecosystem working really well. But I also want to acknowledge that in addition to all the wonderful things the SMA Foundation did, Lauren played the role of matchmaker. And I'll never forget an SMA Foundation SAB meeting where right after we had a dinner, and she sat me right next to Frank Bennett. And it was shortly after that we signed the deal. So thank you, Lauren. But anyway, I want to pick it up where Kathie left off in terms of the clinical development. As Kathie said, we had some data from the open label studies that the patients were deviating from natural history and compared to the PNCR cohort. So we were wondering, boy, could we go to the regulators and get the drug approved based on open label data, or do we have to do controlled trials? And the decision to conduct controlled trials was not an easy one, particularly when parents and many of us were starting to get signs of efficacy. This, as Darryl said, is a devastating disease. It's life-threatening in infants in the most severe form. But after talking to regulators and after much discussion, we felt, boy, this drug is working. We're pretty certain, it's based certainly on the autopsy findings. But the fastest way to get this drug approved and in the hands of as many patients as possible was to conduct controlled trials. So these are the three registrational trials: ENDEAR, CHERISH, and NURTURE. ENDEAR was a study in infantile onset SMA, mostly type 1 infants. And this was a sham-controlled trial, so a third of the patients didn't get the drug, and 2/3 did. CHERISH was a study in later onset, mainly SMA type 2. And again, that was a sham-controlled trial. 126 patients, a third of the patients got sham control. NURTURE was a study in pre-symptomatic infants. So these were infants that were genetically diagnosed as SMA, but still were not with symptoms. Now, you can imagine this is a very difficult trial to enroll in because we had no newborn screening. Actually, one country in the world was doing newborn screening, Taiwan. So we did get some patients from Taiwan. And in the US and Europe, we got patients who were siblings of children of families who had SMA. So we did, because we knew we were going to have trouble enrolling, we conducted an open label, single-arm study. We ended up enrolling 20 patients in that study after screening 25. So this was not an easy decision to make, but we did that because we thought we would get the drug approved as rapidly as possible. I want to show this data. This is some data from just infants now from several studies. What I'm showing here on the y-axis is the mean total motor milestone score in the Hammersmith infant neurologic examination. This was an examination that actually, this scale was developed for infants between the ages of 2 months and 2 years who had cerebral palsy. But we adopted it for this purpose because it was exactly the right age group. And we used the motor milestone section of it. And on the x-axis, we're plotting the visit day, so time from enrollment into the clinical trial. In the lower green line, you see the patients who were enrolled in ENDEAR, in the infantile study, into the sham control group. And the thing to note is that the patients, after a slight increase initially, which may be due to what I would call perhaps a placebo effect, or the fact that the patients were now being cared for at top-notch investigative sites, there's a slow decline. But imagine if the drug only slowed progression of disease. And imagine trying to show a slowing of progression when the progression is that slow. And that's why our initial idea was that we would study type 1, do type 1 trials and look at survival or people who were free from permanent ventilation. So that's the sham control group from ENDEAR. Now look at the purple line, which are the patients who were randomized to receive nusinersen in the same trial. So these are the people, these were the infants who were mostly type 1. All the infants on this graph, on this chart, had a copy number of type 2, so as Darryl said, predicted to be type 1. And you can see that after an initial period where there was no separation from the sham control group, there was a steady improvement. This is unlike you see in any natural history study as well. So this was very encouraging. I want to point out day 183. You can see that day 183 there is some separation, but after a period of no separation. And I want to bring this up because we decided to do an interim analysis in that trial, which led to the rapid approval. In Kathie's slide, you saw that the drug was approved 5 years from first in human studies to approval. That is record time. And one of the reasons for that is that we did an interim analysis in this trial in DEER. That's risky because if the trial doesn't succeed at the interim, now you have to go even longer and you actually risk being positive in the final analysis. And we knew that there was potentially a delay in the onset of action, so the interim analysis had risks in it, but we thought it was important to conduct it because we wanted to get the drug approved as rapidly as possible. In the red line, you see the data from the open label study that Kathie talked about, in addition to the open label extension. And you can see that it turns out that the open label data is very similar to the control data. So our concerns about open label data being out of whack actually were not borne true by the data. And then finally, I want to point to the blue line because these are the pre-symptomatic infants. These are the ones who got drug shortly after birth based on a genetic diagnosis before symptoms. And you can see that they have the most dramatic effect. In fact, many of these children are reaching motor milestones on time. And so far, none of these children, even though they are likely to have type 1, have died or are on ventilation. So I want to then ask Darryl to come back, because it's one thing to see a graph, but it's another thing to see a video of a real patient. So Darryl?
So as Werner Wolf might say, let's go to the video. And let's see if I can do that. So here we have 3 newborn infants who are part of the Nurture study. And they all have a disease-causing mutation in the Smn2 gene. And the question is, what is going to be the consequence of that mutation? And you can see their copy numbers listed there for the Smn2 gene. And so the 2 children on, I guess, your left have 2 copies, and the 1 child on the right has 3 copies. And these are evaluations of these infants at 5 days of age, 7 days of age, and 8 days of age. I would say, just kind of from a personal note, I saw my first patient with SMA in 1960. That's kind of a long time ago at this point in time. And I can't recall how many families I sat with over the years and explained to them that their infant has SMA, type 1, and then had to discuss with them the expectations, which, as you can imagine from previous discussions, was very sad and difficult. And I would say, for me personally, one of the great moments in my career is the thought that I won't have to sit with another parent and give them the bad news that this is a bad disease, your child may likely die from it, and we don't have any effective treatment for it. So what are the expectations here? And you've already heard this alluded to, and I'll take just a moment. But since 2 of the babies had only 2 copies, we would expect in all likelihood that they would have type 1 disease, and you saw that at the very first video. Or alternatively, they may have the intermediate form of type 2 disease, which is still quite disabling, as you can see here with the infant on the right. So what in fact happened with these 3 newborn infants who were treated with nusinersen or Spinraza, and here they are now, between 1 and 2 years of age, running around, developing perfectly normally. The older boy, 25 months old. So that older boy has 2 older brothers, and he's wrestling with them and being very much an active part of the family. So it's a wonderful, exciting moment, I think, in this situation. And I would like to say, as everybody has commented, that there have been critical pieces to putting together this complex, exciting puzzle. But personally, from my point of view, I want to personally thank Lauren and Dinica, who I just don't think we would be standing here today if it weren't for you two. Sorry.
Okay, so we have time for a very few questions because I've been told we turn into a pumpkin at 4 o'clock. So this is a fantastic example of precision medicine, a treatment designed just to patch together splicing in a disease which had promise. I think that the decision to do sham controls was a very difficult one, but obviously the right one because now this is approved. One of the issues, obviously, is if we now know or believe that treatment is most effective before symptoms show, what does that mean for newborn screening? And I don't know. Al, you want to discuss that?
We were hopeful that the results of Nurture would be positive, so we actually started our newborn screening efforts several years ago. It's a state-by-state process. You need to get the test included in the RUSP, the Recommended Uniform Screening Panel, and then even after it's in the and even after a state has approved, and the state of Missouri, for example, that passed through both the state legislature and the governor signed it, it could still take a couple of years before the assay is validated and included in the panel. But obviously, with the data that I just showed and Darryl showed, we really wanted to get this becoming standard of care essentially right after birth if they have genetic diagnosis, and it's a long state-by-state process.
So are there implications for this antisense oligonucleotide treatment for other neurological diseases?
Yeah, I'd like to answer that, is that this is a platform technology, and so what we've learned from SMA we can apply for other neurological diseases, and unfortunately there are a large number of well-characterized neurological diseases that really have no treatments. They're not disease-modifying, and so we're focusing our programs on dominantly inherited neurological diseases initially, so things like Huntington's, some forms of ALS, Alzheimer's, Parkinson's are in our pipeline, and they're partnered with Biogen and with our colleagues at Roche for the Huntington program. But it really is exciting and gives us optimism that we are going to make a difference in treating some of these horribly debilitating neurological diseases.
One of the things we found, Story, was that in the autopsy findings, not only does the antisense get to all the way up to the cervical spinal cord, but it goes all the way up to the brain and affects pretty much all the cells in the cortical mantle. So that opens up a whole number of neurodegenerative diseases.
I'll just say, too, I want to add, so sort of beyond antisense, I think it gives hope for treatment of neurodegenerative diseases, that if you have a targeted therapy that treats the underlying disorder, you can have dramatic effects. I've worked in a lot of neurodegenerative diseases. People always talk about changing the rate of decline. In this case, Al showed from the infants, but in the children, too, we see really dramatic improvement in function, and it gives me hope that that may be possible in other diseases as well.
Stan.
So maybe you could just extend what
Yeah.
you were saying to, do you think this treatment is going to go on indefinitely as the people get older and older? Or is it, when I first heard about this and read about it, I thought this was the immature nervous system was susceptible.
No.
So please comment.
Yeah. So I'll let Darryl comment, too. So these children, as he showed, are not born with symptoms. They're born normal. And then in the infants, they have symptom onset, usually around 3 months of age, but there's a range. In the children, they're actually quite normal until 12 to 18 months of age. So for parents, this is very devastating because you have a child that looks normally, and then all of a sudden you notice something's wrong, and you're told they have this diagnosis. So it's not an immature nervous system. It's actually regression of the nervous system. Your question, though, actually is a really interesting one. Will we have to treat them forever and ever? There is a higher requirement for SMN protein during development, so that requirement may go away, just like the requirement for lots of factors goes away during development. And maybe once they're treated for a while, they will not need treatment. I don't know. We'll have to see.
Another question before we get pulled off the stage?
This is very much similar to Dr. Kruzer's. You know, in terms of the issue about whether something is disease modifying or symptom enhancing, have you done any discontinuation in any of the subjects that have responded favorably?
No.
No.
I don't think the parents would do that.
That's a difficult study to do in the human condition.
Yeah.
But in the animal model, that's been elegantly done by Umrao Manani and Kat Lutz at the Jackson Laboratories. And what they show in the mouse model for spinal muscular atrophy is there's a very high requirement for SMN during the first 17 to 21 days postnatally in the mouse model. And then after the period of maturation of the motor unit, particularly the neuromuscular junction, there's a very low requirement during maintenance of the system. So in the studies they did where they turned the gene on or turned the gene off, depending on the question they were asking and the answer they were looking for, when they turned off the SMN gene at 21 days of age in the mouse, there was no longer a requirement in the mouse model for SMN in order to maintain normal growth and development and full life as such. So we
Adrian,
think that applies in the
Adrian, did you
human
want
in
to
some
say
way.
something?
Yeah, I mean, we also...
And then one last
Yeah.
question from back there, and then we will vacate the table.
With
Adrian.
Ionis, we did experiments where we exacerbated the splicing defect in a mild transgenic mouse models of SMA, and we could show that you could get adult onset forms of severe disease in the mouse. So there are critical thresholds, but it seems like even later it's possible to see these problems if you don't have sufficient levels of the protein. Maybe not just in the CNS, so it depends. There are some complexities that may be unique to the mouse models.
Can I sneak this in? Comparing it to the Fragile X story, which didn't. Are there any methodological lessons or errors that to be pointed out why this was it just the drug is just it was a genetically developed treatment that targeted the gene product and therefore was effective?
Yeah, I think from a molecular standpoint, you would approach those diseases very differently. In this case, we're using an antisense drug to enhance splicing. In Fragile X, you'd have to use a different approach to reactivate the gene. And so from a sort of molecular gymnastics standpoint, I think it's much more challenging to do that. And we heard earlier today that developing therapies to increase expression is still something that the industry needs to do a better job of.
So this was a disease that was uniquely
Poised.
for this kind of very specific antisense oligonucleotide intervention to fix splicing. And there's a question back there. Yeah.
Do you feel that you would have better outcomes at an earlier level if you were able to treat the respiratory distress felt by the infant type 1 patients?
Mm-hmm. Yeah. Yeah, I mean, I think if you can't treat pre-symptomatically, then, I mean, even in our ENDEAR study, we found that the patients who got treated with shorter disease duration before they started getting some of these complications you're referring to, they did better than the people who got treated with longer disease duration.
But I'll just clarify. So the respiratory distress is actually a motor neuron deficit. So it's the diaphragm muscles and their innervation that is affected.
There is a different disease called SMA with respiratory distress as the presenting feature, or SMARD, due to an entirely different genetic mechanism, and it's not what we were talking about today.
Okay. Well, thank you all very much for staying and listening to this an extraordinary success story.
Oh, yeah.